So if you can please take a look at Unit 7, questions 16 to 18. So I'm hoping that the Unit 7, questions 16 to 18 that you're looking at in the red booklet has to do with feathers. Because if you are looking at some graphs that are talking about seals and humans, something like that, then you probably have an old ACER booklet that you downloaded from the internet, or possibly you bought the old booklets. That is possible, and so that's not the current question. So we're not doing a question from the past because that question is no longer being used by ACER. And of course, they have to have their reasons for that. So there's a question about feathers. So if you don't have that question, sorry, there's nothing I really can do. And but we will be doing the next unit 16 probably in about 20 minutes or so, and perhaps that will be of value to you. But I can't post up the question for copyright reasons, obviously, but we are going to perform the work solution. So I'm going to give everybody a minute to look at to remind yourself of this unit if you've done it already, and if you haven't done it, just so that you can get some context to what it's about. But it's a unit about feathers and different colors, blue, green, yellow, like that. So please give it a try. Okay, so looking at the first question, we're asked about the genes for the two pigments and how, and the question is they would most likely. The first answer choice suggests that the genes for the two pigments could differ by one base pair. Now, you have to try to, in the back of your mind, I mean, there's an answer that's clearly correct, but in the back of your mind, you could always remember that, a protein that's made, proteins, sometimes proteins have not so many amino acids together, perhaps 50, 100, but most, usually those are polypeptides, but most proteins are in the thousands of amino acids. And if you remember for some basic genetics, the genetic code is 3 base pairs. So when you think of thousands of amino acids, and each amino acid is coded by a base pair, which is only 3, then if you were to change one base pair out of these thousands and thousands and thousands of base pairs, would that create a different protein? It's just so tremendously unlikely, especially a different protein that works, because it's one thing to have a mutation, but it's a different thing that you're going to have something else that actually is functional in the organism. So it's a very, very unlikely thing. Then the genes for the two pigments most likely be on different chromosomes. So when we look at it, it says in the first sentence that the feather color is determined by 2 independently inherited genes. So that's the key statement. So the genes are independently inherited, and if so, they would be on different chromosomes. So of course, this whole thing is about Mendelian genetics, and so Mendel, came up with these rules, maybe fudged his results a little bit, but anyway, it worked out well in the end. And he has these, 2 primary laws. They'll never ask you what is Mendel's first law or what is Mendel's second law, but this is essentially testing it. But even if you didn't have a science background, that first sentence should give it away. But Mendel's first law is the first law of segregation, And, you know, we have 46 chromosomes. Everybody should know that. And when we make our gametes, you know, sperm and the egg, that has to have half our chromosomes, because if it had our full chromosomes, then when a sperm and egg comes together, too many chromosomes. So sperm and egg have to have half the chromosomes. So they have to go through a reduction division called meiosis. So when before the number of chromosomes are reduced, for example, like a primary spermatocyte, which will produce sperm, that has our normal number of chromosomes, which is 46. Then it has to go through division to become 23. When it is 46 chromosomes, try to remember 23 of those chromosomes came from your mother, 23 came from your father, so maternal and paternal origin. So when those chromosomes are going now to segregate into two different cells, so that you have sperm with 23 chromosomes each, when they're going to move into one side of the cell or the other side of the cell, it is not the case that your maternal origin chromosomes all say, let's hang out together, and all the chromosomes from your father says, let's hang out together and make a new cell. It's impossible. So they sort independent of each other. Few chromosomes go this way from the mother, few that way from the father, and it all mixes together, and that helps to create all the diversity in life. So Mendel's first law of segregation. Mendel has a second law of independent assortment, which is really related to this question, and that's the fact that traits on two different chromosomes will assort independent of each other. Of course, if you have a biomed background, you're thinking about, well, all the exceptions like crossing over and blah, 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 but this is the basic information that they require for you to know for this question. So 16, the answer is definitely B, as in boy, because they said it in the first sentence, basically, so they give you just a little bit of an easy one. But, you know, when you look at C, by the way, be activated by the same mRNA, this is getting a little, it's confused because the principle in molecular biology is that DNA is translated into messenger RNA, and this is produces a protein. So it goes from DNA, messenger RNA to protein. It does not go protein back to mRNA, back to DNA. Yes, there are exceptions in advanced biology, but not at this level. So now 17. What is the expected phenotypic ratio of a cross of that versus that? So what do we have here? We have a cross of big Y, small y, big B, small b, times. Okay, so these are two different traits. If there was just one trait, that would be called a monohybrid cross. Monos for one, and if it's two traits, it's called a dihybrid cross. Though they didn't say it, I'm sure ACER meant that this is random mating, because that's the only way that you would be able to come up with a solution, that this is a dihybrid cross with random mating. Now I remember teaching a course one time, I think it was in a GAMSAT course in Perth or something, and a student came up and showed me this question. It was the first time I saw the question. She was very upset. How could they possibly ask us for a dihybrid cross in an exam where you have to complete the question in 1.5 minutes? So yes, this is really a question that comes up regularly, actually. It comes up a couple of times in ACER's practice materials, and it comes up from time to time on the real exam. So you have to be efficient and observant to be able to complete this in a reasonable amount of time. So first of all, if you have not done this before, I suggest that you try doing it now, the dihybrid cross, and then you can look at the answer choices and see if you can figure out which one is correct. while you do that, I'm also going to start filling in my little thing here. Okay, I guess we've got some different opinions on what the answers can be, B, C, A. So, okay, so let's take a look here. First of all, I would like you to notice that we have what this means, that we have independent assortment, which is what the first question was about, is that this you have these two alleles, big Y, which is dominant, and small y, which is recessive. Big B dominant, small b is recessive. And so this would be called heterozygous for the condition Y, heterozygous for the condition B. Over here, we have two recessive alleles. So we would say that it's homozygous for the recessive allele Y and heterozygous for the trait B. And so now, in terms of what gametes can this individual produce, this individual can produce, because the Y's and the B's are going to assort independently of each other. That means the possible options in this dihybrid cross is big Y, big B. So that's what this is here. This is the big Y, big B. Then if there's big Y, small b, big Y, small b, small y, big B, small y, big B, and small y, small b, which is here. So this represents this organism. This is being crossed with small y and big B. Okay, so we have small y, big B. Boom, that's the first one. Small y, small b, that's the second one. Small y, big B, that's the third one. Small y and the big B. And then small y, small b, the two last ones. So that represents all the possible options. Now we do our Punnett square, and we just are able to put in each cell what crosses that cell. So we have this one is going to be small y, big Y, small y, big Y, then big B, big B, big B, big B, and this one is small y, big Y, and then big B and small b, etc., etc. I don't want to say it all. But you see that each cell represents the two that cross that cell, and that gives us the gamete. Now here is the ACER trick. Okay, normally if they're going to give you a dihybrid cross, one of the organisms will be homozygous for the trait. Now, the advantage of the homozygous for the trait is look at this Punnett square. This part here, small y, big B, is identical to this part here, small y, big B. And this is small y, small b, which is identical to this. Because the top line never changes, you do not fill out this. This will be a waste of your time. This will put you over 2 minutes, guaranteed. If you fill all this out and then start, oh my God, then you have to figure out which cell represents green, yellow, blue, whatever. So you avoid doing that by recognizing that one is homozygous and there is a repeat in the cell. So here is the angle on all this. Doing a dihybrid cross on the GAMSAT in 1.5 minutes is highly unlikely for you to do that unless you recognize that there is a pattern that is developed and you don't have to go all the way. Because will ACER do a 2x2 Punnett square, which means a monohybrid cross? Yeah, it comes up on the exam regularly. But they enjoy this one because it stumps students, and of course it makes it very difficult to finish the exam. But if you notice the pattern, then you realize it's not a 4x4 where you need 16 cells. It's actually a 2x4 where you only need 8 cells that you're going to get the data for. So now the first half of these Punnett square problems, which do come up regularly, is to do your Punnett square and get your results. The second half of the issue is that ACER usually will find a way that makes it a little bit more interesting to interpret the data, so that the data is not just a matter of counting up the squares. So now we have to interpret the data, and by doing so, we look at what they call a green feather. And now ACER is saying anything that has one capital Y and one capital B makes green. So I'm going to look around for one capital Y, capital B, and then I'm just going to, let me see. Okay, so I'm going to look for capital Y and a capital B. And so I find a capital Y and a capital B there. And so that's going to be green feather. So down below it, there is a capital Y and a capital B also together. So that's green. And then over here, there's another capital Y and capital B together. So that's going to be green. So now, what else do they say? They say if you see two small Y's and a big B, that's going to be blue. So we're going to look for two small Y's and a big B. Okay, so I see one here. That's going to be blue. Two small Y's and a big B. That's there. Two small Y's and a big B. Okay, I see one down here. So that's according to the rules. Now we still have two that have not been decided. Now, we know that the Y stands for yellow pigment. That's what the Y stands for. It says it in the first paragraph, in the last line in that first paragraph, or second to last line, that you know, the Y is for the yellow pigment, and you can see it in the diagram. And if you have small b, well, that doesn't show anything. That's just clear. It's white. So all we have therefore is with small b, but a capital Y, which is dominant, that's going to be yellow. And then in the final one with small y, and small b, well, that just produces no color at all. So you see from our diagram, without having to do twice the work, you can see from the diagram we have 3 green, we have 3 blue, we have 1 yellow, and we have 1 that has no color. It's clear, it's white. So that means that for answer choice question 17, the answer is C. I also want you to recognize that another way that they can ask this question is they can ask percentages. So what is the percentage of green? And you would have calculated that it's 3 out of 8 options or possibilities, and then you would calculate the percentage, or you would look at the answer choices to see if there was something just under 50%, and then you would choose that. Depending on how close the answer choices are, you may or may not calculate it. They can ask you, you know, if if there, how many, what's the percentage that are blue, that can be blue or white? And you can just look at it, percentage blue or white, that's 4 out of 8, and that would be 50%. So just to let you know, and that's blue, B or white. So it's just to let you know that there's many different ways to ask the question depending on how much extra work or less work they want you to do. And so this, I think, gives you an idea. And of course, like I said before, this is a case of random mating and a dihybrid cross. Okay, so that's it for 17. 18. So in 18, they say, what happens if this bird, and ACER loves birds, bats, and fish. So they ask, what if this bird has twins? Why not? So fraternal, non-identical twins. So which of the following? So first, you have to have just a little general understanding of twins. You know, it's not that, you know, you have to study this really, because I think an average person understands that non-identical twins are not identical. They don't have the identical genetic composition. So in reality, another way to say non-identical twins or fraternal twins is that these two people were born at the same time, but from a genetic point of view, they're no different than any other brother and sister. So they could, well, I'm not sure if you would use brother and sister in the case of birds, but you know what I mean. So the genetic composition is simply statistical at that point. And just to give you a little bit of background, of course, an identical twin, which is really called a monozygotic twin. Mono means one, and zygotic refers to the zygote, which is the combination of the sperm and the egg, produces the zygote, which is the one cell. And then after that, you get you, you know. So for a monozygotic twin, it's the one zygote that the one zygote that is created. So it's identical genetic information, and then it divides and produces two human beings or two organisms. So that's what happens for a monozygotic situation. But dizygotic, which is fraternal or non-identical, so that means that there were two different eggs. And those two different eggs were fertilized by two different sperm. And it's not that you really have to memorize that or whatever, but it's common knowledge that fraternal twins or non-identical twins are non-identical because their genetics is not identical. And that's all you needed to know for this problem. So here's the good news about this question, is that I would say 80% of the time during the real exam, if you spent a lot of time trying to solve a problem, like creating this chart that we just did, if you spent a lot of time creating a problem, there's a high likelihood on the GAMSAT that there's going to be some sort of follow-up question that's going to be easy, but based on the hard work that you did. So that's the good news. And so this question, you might think it's independent. It's completely not independent. You work too hard for that. So the first answer choice, it says, is that the offspring cannot have the same phenotype as the parent. Well, let's see what we learned from the previous question, question 17. We saw this cross between two. Well, big Y and big B made. Oh, I guess I can add some color to that as well. Okay, so I just added some color. Going by the rules that ACER provided, big Y, big B produces green, and small y, big B produces blue. So we know that green and blue make all of that. And now, with that information, we look at the answer choices for question 18, and it says the offspring cannot have the same phenotype. We just see here that the parents have green and blue, and they have offspring with green and blue and other colors. So answer choice A is incorrect. The offspring cannot have the same genotype. Do we have any genotype that's identical to the parents? Yes, we have this genotype identical to the genotype of the potential offspring down here. So we know they can have it. So answer choice B, we can also say is definitely incorrect. The two offspring and the two parents could not all have different phenotypes. Could not all have different phenotypes. So phenotypes is what they look like. By the way, genotypes is the genetic composition, which is the big Y, small y, big B. But the phenotype is what does it look like? What color, for example, is it? And is it possible for the two offspring to have both to have different phenotypes than the two parents? Of course, because we have green and blue as the two parents, but it is possible that one child is yellow and the other one is white. So that is possible, and that means that they have different. So doing that diagram permitted you to answer question 18 quickly, faster than what I'm doing because I'm explaining each one, of course. So, and then answer choice D, the offspring could have the same phenotype and genotype as one parent. I'm sorry, I have to add some time. Okay, so the offspring can have the same phenotype and genotype as one parent, and we saw that already. Big Y, big B, small b. Big Y, big B, small b, and it's green, So same phenotype, same genotype, and so answer choice D is correct. And by the way, some students will choose answer choice C because of the double negative in the question. And so when you see that not or accept or whatever during the real GAMSAT, it's your test, you paid for it, annotate, circle, underline it, put a star in the margin so that you continue to work that through in your mind so that you don't confuse the wrong answer with the right answer. Okay, so that's it for Unit 7. We're now going to move on to Unit 16.